Okay, so time's up, and I think we can just start the second session of today's colloquium. In the second or the last session of this colloquium is more focused on environment and One Health. So in this session, we have three wonderful speakers with a distinctive background of expertise on different aspects of One Health. As you can see in here, the first talk of this session starts with a case studies showing animal, human, and environmental interface. And the second talk is more about interaction between inland water and human health with an emphasis on harmful algal bloom. And the last talk is more focused on forest, the wood and forest and biodiversity in the context of public health. So in this session, we have so many different aspects of environment, and you will see how these environmental aspects are closely linked with either human and animal health. So let's start with the first lecture. The first talk of this session starts with an animal, human, and environmental interface, as I mentioned. And Dr. Schneider will show her expertise and experience in Latin America to show her health hazard in this interface. Dr. Schneider, the first speaker, is a professor at the Department of International Health at Georgetown University. And she has a veterinary medicine training, but also in public health education. And you'll see she has a very unique work experience in government of Mexico and also international organization of WHO, and now with academia on this specific topic of One Health. So we are very happy to have you, Dr. Schneider, and please join me welcome Dr. Schneider with a big applause.
Thank you very much first for the invitation. Thank you to the Seoul National University, the College of Veterinary Medicine, the organizer of this wonderful collection in One Health and Environment. It's a pleasure to be here today. My presentation was, as mentioned, is a little bit for different experience because I have been in the interface for a few decades now, working from local to national to international organization, and now I'm at Georgetown University. I'd like to start my presentation. I mentioned that it's so important to respond to the current challenge that the world is facing now to understand the link between humans, animal, and the environment. And was mentioned for the previous speaker, how important is to work in multidisciplinary collaboration. We already know for 20 years that 70% of the infection hazards that threat public health are in the interface with animals. Several examples was given today, like the SARS that everybody talking about, but also Ebola is in the interface, and influenza outbreaks and pandemics time to time are in the interface. And we'll not forget rabies was already mentioned, and plague that still persists in some countries in the region. We are 8 billion people traveling very fast and easy around the globe. We have very high rates of urbanization, also very high rates of poverty, and many conflict, migration, climate change, so many challenges that the world are facing. Professor Salomon did a wonderful introduction about the One Health history. I don't need to repeat. I only would like to include 2 examples for the American region that I was part. First was the rabies surveillance system. In PAHO WHO, PAHO is the regional office of WHO for the Americas, and it was established the veterinary public health program in the 40s. In the rabies surveillance system, that rabies was a challenge in that time, mostly for Latin America, is from the 60s and was in the beginning with this integrated vision. It was reported human cases, animal cases, domestic and wild, And Ministry of Health and Ministry of Agriculture report data. I was part of the rabies program, the Ministry of Health of Brazil in the 80s, the beginning of the 90s. And my thesis and PhD and master was on rabies. But was in 2004, as what mentioned, they had a meeting in New York that started using the term One Health. And that was just start SARS-CoV-1 and have many outbreaks of influenza H5N1 that affect original Asia, but many other parts of the region. In that time, I was already working in PAHO WHO in the headquarters in Washington, DC. And we started working with the countries. I was the Department of PAHO Health Emergency. And we did many meetings with the country representatives, health and agriculture, to do preparedness plan integrated in collaboration, health, agriculture, defense, environmental, depends of the country's structure. And this was very useful for when arrived the influenza pandemic H1N1 in 2009, that the country was already talking with each other, different sectors. And as I mentioned, many things are going on, like international meetings, and the framework of FAO, OIE, doesn't stop anymore to see the importance of One Health. We wrote a chapter for Oxford Encyclopedia of Global Public Health about One Health. And I have the pleasure to have Dr. Kyung Do Min from this university as co-author. And we wrote a definition, a transdisciplinary definition for One Health. We think that it views how humans, animal, and they share environment, like such ecosystem, are linked and could be also affected by the human interests, such as food production, and have pressure, external pressure, such as urbanization. And we also consider that different disciplines can together provide new methods, tools for research, implementation of service to predict, prevent, and respond to events in the interface. This is open access, this chapter, who wish to see more examples for the American region. I like to use the One Health component to my examples, because this is from a meeting some month, and it happened some years ago, but it's very easy to give the perspective of One Health. I will present two studies in leptospirosis now. And the human component, we could suggest that's close to 1 million cases every year globally. In the American, 10,000 cases annually. And the people are usually exposed to the urine of infected animal indirectly through water or mud. Could be occupational hazard, could be a recreation hazard, and like 10% of fatality rate. And the animal component, most of the mammals could have the leptospira. It's well known the relation with rodent, domestic and wild, but also cattle, and they could have economic loss. There's a lot of abortion with cattle. And also you can have other animals, horses, dogs. And the environmental component is already known that it's a disease most common in the tropical countries and after flood. And I will present a few other examples of the environmental component for leptospirosis. The first study was made together with Nicaragua, with the government authorities, the Minister of Health, Minister of Agriculture, and also the University of Nicaragua, and a multidisciplinary team at PAHO. And that was mentioned for the previous speaker, the importance to work in collaboration. We have in our team a geographer, a biostatistician, veterinarian, physician from different institutions and different disciplines. And Nicaragua is a country in Central America that have two oceans, Pacific and Atlantic Ocean, and is very vulnerable to natural disasters and have been facing leptospirosis outbreak for several years. They have a very good surveillance system in the country and an intersectoral plan, health and agriculture, together with the support of university. And the objective of this study was to stratify the risk areas of leptospirosis in the country and also explore possible drivers. As the methodology, we choose an ecological type of study using aggregated data by subnational, second subnational level, was municipalities, 153 in Nicaragua. We use surveillance data from the country, time period. Also, we use information from the census and many other environmental variables. It was a total of 15 variables in this study. And we also did exploratory analysis using GIS and also statistical model. As results, this is the risk map of Nicaragua. For this risk stratification, we use the criteria of critical areas like hotspots, upper quintile of cases, and rates. And this is the red ones is the critical areas for leptospirosis. Yellow is the endemic area, and the grays is the silent area. Now we need to check if there is really no cases or need to improve the surveillance system. Nicaragua in that period have between 60 to 600 cases every year. Depends mostly of the weather that I will demonstrate very, very soon. The most important finding of this study was the relationship of cases of leptospirosis with the type of soil. And this is Nicaragua. You can see that this country and many others in Central America have a lot of volcanoes. And this type of soil, orange in volcanoes, it will have a higher association with leptospira. And talking with the experts in this bacteria, we understand that the bacteria like certain types of pH. And this type of soil that we have a geographer in the team, keep the bacteria longer surviving. And then could be like a place that you have longer, for more time, the bacteria and more cases. The other environmental variable that found the association was with precipitation. This is not new. We know that leptospirosis is many times after flood. This is a figure with precipitation by months. And the next month cases of leptospirosis, and after heavy rain, we have a concentration of cases in the Pacific area. And also the other variable was the percentage of rural population. Nicaragua have 50% of rural population, and after heavy rains, that is constantly flood for several days. And also the population, this is a hot country, use the rivers and creeks for recreation and also washing clothes or to have a bath. This information was used by the decision makers because they are part of the study, they are part of this analysis. And the recommendation that if we recognize where are the critical areas, what municipalities or county are the critical for leptospirosis, in what period of the year you have more cases, you can concentrate your efforts in these areas in that period. And then could be used for rodent control before the heavy rain, could be used to alert the physicians that is leptospirosis time, this is the test, this is the antibiotic, you can use this for the decision making. And Nicaragua share borders with Honduras that have the same environmental drivers. And then we work also with Honduras to help them to be ready to respond to outbreaks. And this was a simulation exercise, health and agriculture together, the 4 levels from local to international. The second study that I would like to share with you was done with the state of Rio Grande do Sul in Brazil, my original state, and was also a mix of institutions. We have the state Health Secretary, Agricultural Health Secretary, and also the university and a multidisciplinary team. As was mentioned, I'm a veterinary doctor, epidemiologist. Patricia is a geographer. We also have a biostatistician. We have physicians. We have a multidisciplinary team. And leptospirosis is a disease of mandatory notification in Brazil. It's part of the surveillance system. Any case of leptospirosis that we saw in our health system need to have a notification to the surveillance system. It's around 4,000 cases annually that it's reported to the system, 10% of fatality rate. And for my original state, is important because the economy is based in agribusiness. We have a lot of plantations and cattle raising. And the objective of this study was to analyze the distribution of leptospirosis and also explore the possible drivers and give recommendations to the decision maker. We use the same style, ecological type, using aggregated data by subnational level. That is close to 500 municipalities in my state. We get a data set from the surveillance system, from the Ministry of Health. Also, we use data from the census and many other different environmental, socioeconomic data. You can see some that we use, some say for the final model, others not. And we create a database from scratch, folded, and we did also some statistic analysis. I think we used 15 variables. This is already the results. This is the state of Rio Grande do Sul. The concentration of urban cases are here in Porto Alegre, when I am right now. I'm in Brazil right now. This is the concentration of cases, urban, but we have two big areas of concentration of cases in this area close to the rivers and in this area close to the lakes. It's around 400 cases reported every year. But the variable, the environmental variable, more interesting in this study was the ecoregion. You can see this green area, that's Paraná, Paranapaíba rainforest. This was with the strong association with leptospirosis. You can see the concentration of cases in the green, the ecoregion. But also we find some association, not so strong, but we found some association with a production of rice. This is not new. It's known that many paddy rice workers have leptospirosis. The new finding was the association with tobacco plantation. And trying to explain this association, I think that's related to the pH that the plant, the tobacco plant like, is the same that the leptospira like to stay longer. And also the way that the workers deal with the leaves, they touch a lot the leaves. In this study, we also find association with the type of soil, not volcanoes type of soil. We don't have volcanoes in this area, but other type of soils. And as we are working together with the culture sector, we have information of the number of bovines by property in the municipality, in the county. And in the univariate analysis, we found some association with small property, up to 10 bovines. But in the final model, the multivariable model, the association disappeared. And as a conclusion, it's a very important problem for our state now, and it's important to work together because many of the things that we saw here was related to agriculture, are related to the environment. This is important to work in collaboration. And we present this to the decision maker. They are part of this study, but then the government changed, the high authority changed during, after we finished the study. We prepare a policy briefing to the new authorities with this study and two others that we did later to show how important is this topic, and for this to be only a publication, to be something that could be used to save lives of leptospirosis. These two studies was done when I was working at PAHO. Now I worked almost 20 years at PAHO. And after I retired from PAHO, that I joined Georgetown University, and also I collaborate with the Federal University of Rio de Janeiro. I changed research topic, and now is snake bites. And only to show you this components of One Health approach, looking in snake bites hazard. In the human component, is close to 3 million people that are bitten by snake every year around the globe, mostly rural population. Could be occupational risk. It's estimated that 100 people annually die by snake bites, and many people have disability. And 5% of fatality rate if you don't arrive to a place that have the antivenom in a short time. In the animal component, is close to 3,000 species of snake around the globe, plenty of medical importance. Animals are bitten also and have economic loss. And the environment, we can see that there's a tropical forest and certain type of bioma. This is our more recent publication now on the snake bite with One Health approach. And I have the pleasure to have Dr. Kyu-Min as a co-author again. And this is our suggestion of One Health approach for Brazil in snake bite. But I will present this study that is an overview of snake bite and possible driver and a risk map. And again, Dr. Kyu-Min as co-author. It's a pleasure to be collaborating with this university for several years now. As a background, in Brazil is close to 3,000 people that are bitten by snake in Brazil every year. It is mandatory to report to the surveillance system. And the antivenom is free of charge. The government provides the antivenom for the people bitten that arrive in the public system. But there is a shortness of the antivenom globally. For this reason, it's very important to know where are the higher risk area to have the antivenom where it's more needed. And again, it's a multidisciplinary study. We have different disciplines working together at different institutions and 3 different countries. And it's really a collaboration of different countries and different disciplines and institutions. The objective of this study was to provide an overview of epidemiological situation and snake bites and help the decision makers now with this tool for analyze risks and also understand the drivers to include in this tool. We use the same model, ecological type of study using aggregated data by the second subnational level. And the surveillance system from the government. Also, we use several environmental variables from different open access sources and demographic and socioeconomic from the government sources. We also create a database from scratch and use GIS and socioeconomic and by statistic analysis. As result, here is the distribution of snake bites in Brazil. You can see that there is snake bites everywhere in Brazil. Most of the municipalities have cases of snake bites, but you can see a concentration of cases in the state of Pará, in this region. And this is all Amazonian legal region. This is forest. And you can see this cluster analysis, the high municipalities, not the municipality that is high and the neighbor is high, is all the legal Amazonian region of Brazil. And also this is forest too. This is forest and this is Atlantic forest in the coast of Brazil. And as a result of the statistic analysis, This was one of the variables that was more important as a result of the study, is the relation with bioma. You can see in green that forest, not tropical forest, is a concentration of cases. Now, forest here, forest here. But also 70% of the cases was in the bioma related with forest, but 60% of our territory are forests. And that means that this is a public health problem, and snake bites is relevant for the government. It's really an important study. This is the variables that stay in the final model. This is temperature, this is precipitation, also elevation was significant. And these two variables are protective effect, like urbanization, more urban areas, less snake bites is affected. No? And also the GDP per capita, more rich areas, less snake bites. But this was the variable that was more relevant to the study, in my opinion. It's not the one more significant, but was significant, but it's this map shows so much. Now here you can see the map, this is 3 laws. This is the deforestation. And you can see the concentration of snakebite cases in area of deforestation. This is a very important finding for my country. And the other map is snakebite richness. There is the overlapping of snakebite venom snakes, the genus of venom snakes. And this is the risk map that we create a tool. I like to do tools of risk. This is for rabies before now Leptospirosis, and now snake bites. And we use epidemiological data, again, higher quintile of cases and rates, but also we include environmental variables to define the risk, including this in this tool. And this is, again, my stake, because I still collaborate with my previous colleagues many years ago, but I still in contact with them, and they are part of this study. We have co-authors from the Ministry of Health. My previous colleague that distribute the antivenom is part of this study, and my colleagues in my state that distribute the antivenom are part of this study too. And we found that most of the Rio Grande do Sul state is medium risk. And we believe that this is something that everybody agrees that we are in medium risk. And it's important to understand that the higher risks are in the Amazon region, mostly forest region. And this is a difficult access area. Now, many times the access to the closer city that have a hospital with the antivenom is by boat and take longer to arrive there. For this reason, it's important to understand this mechanism and see also other type of antivenom that's easy to don't need refrigeration. This is my last slide. I would like to suggest some ideas for the interface post-COVID. The first suggestion is to have this integrated vision. Now there is a name of One Health, but we have been working with this integrated vision for many decades. Since the veterinary public health departments and in the Ministry of Health of Brazil, we are working since the 80s, since the 90s with this integrated vision. And we need to understand that people, doesn't matter where they live, is in Brazil, in Europe, in Asia, they are in certain time connected. We can see now with COVID-19 how fast it spread this virus, no? And we are connected with animals and we share our environment. For this reason, it's so important to preserve the natural habitats and avoid the spillover of new viruses, of new diseases. Abolish wild animal trade carry out surveillance is very important. We need to know what's going on. But if you have a biosafety to do this, if you have a secure laboratory that you can bring from the wild samples from the virus or diseases that could be high lethality, and observe biosecurity. Biosecurity in the food chain, good practice of manufacturers, practice animal welfare, avoid wet markers, and also biosafety for the employee. But this is what very, very brilliant measure mentioned for Dr. Salomon, all the importance of biosafety and biosecurity. To the employees use PPE when it's necessary and be vaccinated against influenza, who work with the production of poultry. This is many things that was mentioned before. This recommendation, number 6, define the priority zoonosis, is not exactly my recommendation, but I have been in West Africa 3 times with WHO in a joint evaluation committee that is expert for other countries that work with expert from the countries with the joint evaluation of the country core capacity to respond to international health regulation. And one of the questions we do for the country's authorities, if they have a list of priority zoonotic diseases, and if this list have agreement between the animal sector and health sector, and when it's possible environmental or other defense, who is part of any emergency team in that country. And these priority zoonotic diseases need to be shared information. You need to have systems and ways to the people who is in charge to share this information. Have meetings now constantly, do joint evaluations, because when you have an outbreak in the interface, you already know each other, you know to who contact, you already know the baseline of the information of what was mentioned, how important it is to have information. And to know if something is unusual, you need to have the baseline, and then you need to share the information about sectors. And you need to continue doing transdisciplinary studies, training. No, this is very important. And my last recommendation is solidarity. Solidarity in all levels, inside your institution, in your country, among countries. We only will have a better world, a more safe world, if you have solidarity among us. No? And this means to have less social inequality and look for a sustainable environment for our generation and for the future generation. This was my last slide, and I really want to say thank you and muito obrigada in Portuguese. And I would like to share a few pictures. This is Georgetown University. This year we are teaching in this building. It's the older one, more than 200 years. And this is the regional office of WHO in the Americas. I spent 20 years in this building. It's in Washington too. The university is in Washington, the PAHO is in Washington. And this is my vet school, the Federal Rural University of Rio de Janeiro, my university that I went to study veterinary medicine, and it's the older one of Brazil. It's more than 100 years. It's close to Rio de Janeiro. Thank you very much. It was a wonderful opportunity to be here. Thank you very much, and I'm available to questions or comments now or later. Thank you.
Okay, so thank you very much, Dr. Schneider. I think the integration of vision that link environment and wildlife and humans are very relevant, especially in the time of climate change. And I think many things could be put in that context. And in that regard, I think your lecture was very insightful. And I hope that this opportunity could bring us to more solid and stronger networking collaboration with your existing link with SNU. And for the sake of time, I think for everybody who has a comment or questions, use the chatting board for her. And I would like to thank again for your great lecture. Okay, so let's move to the next lecture. Our second talk is about interaction between inland water ecosystem and human health with an emphasis on harmful algal bloom, HAB, which is highly relevant in Korea too, because we are suffering from that kinds of issue every year these days. So I think many people will enjoy this lecture. And Professor Brian Brook, actually a little introduction is necessary in here because he is very famous guy, especially in our department. He has been working very actively on water poisoning, water pollution, and also comparative toxicology, environmental health. And he's now serving as a editor-in-chief in a very highly respected American Chemical Society journal, Environmental Science and Technology Letters. And today, we are very happy to host him to share his research on this harmful algal bloom in connection with human health. So please join me welcoming him with a big applause. Brian, it's your turn.
Thank you, Professor Choi. Can you hear me okay?
Yes, very good.
Well, it's a pleasure to be with you virtually. I've enjoyed my previous visits and lectures at Seoul National University, and I look forward to opportunities to engage between our institutions in the future. As some of you may know, I work very closely with a recent graduate of Seoul National University and part of our research team. And some of the things that she's working on is related to the fairly potent toxins that are produced by cyanobacteria. And of course, this is an interesting title. And so I think as we go forward, I'd like to explore a question with you. Let's start with, are harmful algal blooms becoming the greatest threat to water quality? Maybe controversial type of question. But as we go through the presentation, I would submit to you that in some places, in some cases, and at some times, the toxins produced by these HABs are already the greatest threat to people and ecosystems. And of course, harmful algal blooms is a very classic One Health topic. Very classic One Health topic. The forcing factors include things like climate change, as Professor Choi was indicating, landscape modification. I think about the types of observations that Dr. Schneider was presenting. Think about even in Brazil, other parts of Latin America, but harmful algal blooms in inland waters that we use for drinking water, for recreation, for fisheries, are common all around the world. And with the prospects of climate change and continued landscape modification, the forcing factors exist for this to become an even bigger concern in other locations. It's probably not surprising that we've seen harmful algal toxins and the blooms in general being identified consistently in different locations through something called global horizon scanning, which is identifying important research needs related to environmental quality in different parts around the world, not just coming from academics or from one discipline, but from multiple disciplines and business and government sectors in different locations. Here are the outputs from global horizon scanning thus far from Europe, North America, Latin America, Oceania, and more recently Asia, with the efforts from Africa in the works. It's probably also not surprising that we see these interconnected questions related to harmful algal blooms that really represent the topic we are here today to examine, One Health. Because in fact, focusing in just one discipline or in another discipline will never allow us to be able to understand, engage, and intervene when it relates to things like harmful algal blooms. So we see these types of questions that emerge from global horizon scanning, where we simply ask people all around the world what they thought some of the key research needs were to achieve more sustainable environmental quality. It's probably also not surprising that through our recent efforts with the Centers for Disease Control and Prevention's National Center for Environmental Health here in the United States, we undertake something called Uncover EH, understanding needs, challenges, opportunity, vision, and emerging roles in environmental health. The first time that we've examined and learned directly from the environmental public health workforce in health departments in the United States in a concerted and formal manner since the early 1960s. And this was a key partnership with the National Environmental Health Association. We hope to be able to extend this model to other places outside of North America, because the benefits include maybe some surprises that health department personnel are now dealing with the toxins of harmful algal blooms, because perhaps in the United States, states and federal agencies are not necessarily taking this on. But let's think about water quality. Okay, let's think about water quality. Well, what is water quality? Water quality probably means different things to different people. Maybe there are pathogens, maybe there are chemicals, there are different types of pathogen, bacteria, viruses. Think about the rise of Legionella in the United States over the last 10 years with our drinking water supply, and even the emergence in locations after lockdowns in response to the COVID-19 pandemic. But when we think about water quality, I am always reminded by what one of my graduate mentors used to say: you only find what you're looking for. Monitoring surveillance systems are necessary. You've probably also heard that if you cannot measure it, maybe you cannot manage it. So let's think about this principle of water quality as it relates to the types of surface water quality assessment and management frameworks that typically exist around the world for aquatic concerns. Well, we have an assessment framework, and this involves some surveillance and monitoring activity. And we're looking at using the indicator approach. So we select different chemical constituents, different types of pathogenic indicator microorganisms, and then this is what we're examining in our source waters, in our drinking waters, in our recreational waters. In the United States, as just an example, we have hundreds of reservoirs, well over 400, just managed by our Army Corps of Engineers, that provide drinking water, water for agriculture, recreation, and also fishing activities, where some people depend upon the fish as part of their diet. But in fact, we don't really have a good sense for exposure to algal toxins in these freshwater bodies across the United States. As just an example, it's even less understood around the world. What about the different uses, like swimming or fishing? There's evidence that some of the toxins are making their way into agricultural crops that are using waters from the surface water bodies affected by inland harmful algal blooms. And what about toxins in aquaculture? FAO indicates that we need a 200% increase in food production by 2050 in low and middle income countries to meet global food demands. Aquaculture is going to become incredibly important. The blue revolution is going to help us feed many hungry mouths. How often are we examining toxins from some of these substances, including those that may start in freshwater bodies and travel along a freshwater to marine salinity gradient into coastal systems that oftentimes are perfect for various types of aquaculture activities, including shellfish? In the United States is just another example. We have thousands and thousands of smaller water bodies. Oftentimes these are on private lands. Some of these are managed by various government or state level operations. But just to illustrate, this is the Trinity River watershed in which the Dallas-Fort Worth metropolitan region is located. I'm located just south of this watershed. But the Trinity River watershed is critically important because as you go downstream, the Trinity River is impounded by Lake Livingston, which provides a significantly important drinking water supply for the city of Houston, Texas. So we have two of the largest and fastest growing metropolitan areas in North America that are linked by this one watershed. And throughout the watershed, there are many, all of these red dots are these very small, relatively small ponds and lakes. We're unfortunately not examining things like swimming and fishing and agricultural uses and aquaculture activities. In fact, it's such an honor to be with you today with the College of Veterinary Medicine and the School of Public Health. I might submit to you that historically in the state of Texas, the only monitoring and surveillance for algal toxins produced by cyanobacterial blooms would be at a state veterinary examination office when cattle and horses die each summer because they have consumed toxins produced by cyanobacteria. We use various types of approaches in our laboratory, including targeted analyses for some of the molecules you may have heard of. If you have not, but you're interested in One Health, this may be a subject of interest to you. When some of these toxins have nicknames like very fast death factor, this is of concern to both companion animals, agricultural crops, right, and humans. These are very potent neurotoxins, hepatotoxins, and quite frankly, we're even learning that focusing on one individual congener may underestimate reproductive and other adverse outcomes that result from understudied inland toxins. There's even evidence that retinoic acid-like compounds are being produced during some of these cyanobacterial blooms that are teratogenic at environmentally relevant levels. And to examine some of the understudied substances, we also use non-target analyses. And so these accurate mass types of approaches are critically important, oftentimes providing opportunities to understand not just the occurrence and estimate the exposure to people or aquatic ecosystems or terrestrial wildlife, but also to be able to think about the fate or the breakdown products. Some of the work that we're doing now is examining how environmental conditions or gradients in the environment result in the production of toxins and how that may vary in time and space. We also will be examining how when some of the source waters that may contain these substances are treated for drinking water, the development of disinfection byproducts, which can be very concerning, emerge. And this allows us to start fingerprinting and understanding differences among groups that have strongly different biological activity profiles. This then allows us to prioritize other substances that may be of interest using forensics types of approaches for more advanced investigation. But again, the first step in the way that we assess and manage water quality in many places around the world, I would say most places around the world, are not examining the presence of the harmful algal bloom species and certainly not the toxins that may be produced by them that presents risk to animals, humans, and other aquatic and terrestrial wildlife. Yet we know if we can deliver things like the essential services of environmental public health, especially at the local level, we can actually protect and promote public health. Yet monitoring and diagnosing is very challenging, as you may imagine, because we only find what we're looking for, and we may not be looking for these types of threats. And so the scheme, again, that is used around the world is that if we perform this surveillance and we identify a problem, how do we do so? We compare what we observe to some sort of a standard, some sort of a guideline, some sort of a numeric or narrative indicator of a chemical or a microorganism of interest. So again, we use a water quality framework typically around the world that depends upon the indicators. We can't afford to measure everything everywhere, right? All the time. It's just not possible. So we develop the indicators, and an example in the United States are what we call the criteria and the standards. So the criteria, these are recommended and estimated concentrations. The standards are more legally implementable limits. So when we think about water quality, it may mean different things to different people, but in fact, the indicator approach and the numeric values become really important for us to be able to identify if some impairment to use for potable water supplies, for agriculture, for fisheries, for recreation has actually been affected. Yet we see the fish kills every year. In my backyard, in your backyard, around the world. And unfortunately, we don't have that many criteria and standards that have been developed beyond some of the general guidelines and screening values. And this is only limited to a handful of the cyanotoxins. You may be familiar with, for example, more standard 1 microgram per liter levels for microcystin established by the World Health Organization. It's more complex than that. I can tell you that in local reservoirs used for drinking water, recreation, and fishing right now, we reported to a state agency yesterday the presence of multiple cyanotoxins, one at very high levels, and neither of those substances are a microcystin. Well, but the framework, again, if you implement it, can be effective at protecting and restoring the quality of the water supplies. Because again, if you have the information, you can compare it to using the indicator approach to values that are protective of public health and the environment. And when you identify locations that have been impacted, then what you can do is you can actually prioritize where you want to implement your management activities. Of course, with climate change, we're going to have to think creatively about adaptive management approaches, especially as we start identifying locations that are most susceptible to the development of harmful algal blooms and how that may affect things like biodiversity, drinking water quality, and even our aquaculture operations. But again, the challenge is just an example in the United States. The designation of a water body impaired because of a harmful algal bloom is pretty rare, and it really differs among and within countries around the world. So our system can work, but unfortunately, this results in what I would suggest is no restoration, right, of the impaired water quality for HABs is often occurring because our system is essentially broken. We see harmful algal blooms as a transformative threat in some places, in some cases, and at some times going forward. Because the forcing factors, when we think about systems-based approaches, are only apparently getting more pronounced. And we see headlines in our backyard all the time. It was so nice to see a dog, okay, on the announcement for the forum today. I like my dog, I must admit, Professor Choi. and my children like our dog. Yet we see every year, including this year, companion animals dying, right? And we're seeing an increase in many places around the world when we are looking in the magnitude, frequency, and duration of some of the blooms. We oftentimes think about these issues in my part of the world as being a summertime phenomena. Yet the algal issues started here in Texas much earlier in the year than when we might expect. And here's just one example of antitoxin A. It's a neurotoxin. It's produced by a number of species. It's a small molecule, and you don't like it above certain doses. Neither does the companion animals that are going into these reservoirs and actually perishing largely from the antitoxins based upon what we and many other people are observing. But we can start thinking about locations. We can start thinking about regions around the world where things like drinking water and recreational guideline values may be more likely to be observed. Unfortunately, however, when we start thinking about the implications, the historical literature on some of these types of molecules is very limited. Now, we're going to talk about toxicology for a few more minutes. The standards availability of the pure compounds has been low. In fact, only about 5% of the previously published studies on this compound even identified what the purity was. And so there's some major challenges with improving the experimental observations going forward. But one of the opportunities is really taking more of a comparative biology approach. So when we think about One Health, I think about comparative pharmacology and toxicology. And in fact, if we think about the explosion of genetic information among species just over the last 15 years, things like sequence alignment to predict a cross-species susceptibility from the United States Environmental Protection Agency is an example of one incredible tool that is identifying how targets, okay, are evolutionarily conserved across species. We will see this continue to move forward because efforts around the world to replace the use of vertebrate animals in toxicology, in safety assessments is increasing. We will also see around the world more of a move to alternative models. And what will support these types of activities is the evidence based upon the conservation among different species. And so, in fact, we would put forward for your consideration that comparative pharmacology and toxicology is an interdisciplinary and integrative approach for One Health. It presents a decided opportunity for us to think about some of the broader challenges, some of the biggest global health threats associated with the increasing chemical pollution in many places around the world. Of course, oftentimes experience the greatest in low and middle income countries. We can use things like fingerprinting chemical activity, which is coming from pharmaceutical development, to identify chemicals of concern, including some of the harmful algal toxins. And you see our broader workflow here where we look at pure compounds, but we also look at cultures across environmental conditions to identify not just how the substances that we know may affect different adverse outcomes, but also how the culture conditions representing environmental complexity in the natural world results in adverse outcome pathways that can be useful to understand across species implications. Here's just some of the work that Leah Levin, one of the students working with us, has been doing with that antitoxin A molecule. We're also probing ecological stoichiometric types of hypotheses to help us develop better predictive tools. As it turns out, a number of these algal toxins are nitrogen rich, and thinking about nitrogen to phosphorus stoichiometric conditions may help us understand what is actually happening in some places, in some cases, and at some times. This field of stoichiometric ecotoxicology has been put forward this year as a new subdiscipline to understand how stoichiometric or nutrient differences influence accumulation and toxicity of chemicals, how some of the stressors can influence nutrition of organisms in the field, and also how the water quality conditions and stoichiometric differences influence toxins production. And so if you're interested in harmful algal blooms, a recent synthesis paper coming from one of the NSF-funded Nimbios workshops by Angie Peace and colleagues I was grateful to support, has just put forward this as a new subdiscipline. Because the implications is not how many cells we count of a harmful algal bloom causing cyanobacteria or algae. It's the conditions that actually cause the physiological stress to the organism that modify the actual amount of toxins that is being produced per unit cell, and thus the subsequent risk to people and animals. In fact, what we're doing now is developing with our colleagues here and elsewhere better forecasting tools to be able to understand across the complex of gradients when, where, and how risks to people and animals may result. So again, are these harmful algal blooms becoming the greatest threat from a water quality perspective to public health and ecosystems? It's very debatable, but in some places, in some cases, and sometimes they already are. Thank you for the opportunity. I would just leave you now with some suggestions for research needs. We need the better predictive models so that we can scale across time and space. Development of better sensors, standards, criteria, and adaptive management interventions are going to be important as we think about the increasing threats of harmful algal blooms to people and ecosystems and companion and agricultural products. But we absolutely need engagement across disciplines. This is why I'm so encouraged by what you're trying to do at Seoul National University to think together across the disciplines. And it's just been my honor and pleasure to be with you here tonight. So I want to thank you for your attention. I want to thank the team of faculty and postdocs. And there's a graduate of Seoul National on the bottom, Professor Choi, that you know, and students, and also our research funding from the National Institute of Environmental Health Sciences, Oceans and Human Health Initiative in particular, and the National Science Foundation. Thank you very much again for the opportunity.
Okay, thank you very much for your very interesting and insightful lecture. Actually, frankly, I have been reading a series of papers published by Professor Brian Brooks, and I learned a bit and bit from your previous studies. But when I was listening to your lecture, I was thinking again about comparative toxicology. I mean, I would put it like evolutionary toxicology in One Health context, because we have been evolved by interacting with our environment for like millions of years. And then during that time, we became Homo sapiens, but we also see our kind of like, brothers and sisters in different species in ecosystems. So many things are shared in common, and those things can be a subject of toxicology, but also a subject of like medicine, veterinary medicine, public health. So, I mean, this is the insight I got today, and maybe you just only mentioned algal bloom, but I mean, based on that, this lecture, I mean, at least me could be thinking about the big picture or context with which we can envision the One Health area in a language of I'm familiar with. So, I mean, I do have some questions, and I see some questions in comments in the chatting board. But for the sake of time, I think we have to move to the next lecture. But before we move, if you could share the paper you mentioned, stoichiometric, ecotoxicology, that should be really nice if you can share in this chatting board. Okay, so yeah, please join me appreciating what he has delivered today. Brian, thank you very much. Okay, so our last, but to me, the most important lecture is waiting. Professor Dr. Min Kyung-duk. He is a proud graduate of Graduate School of Public Health, and he has been working on areas of human and animal health together. His talk is more focused on forest, ecosystem, how do they in line with public health. And as you can hear from Professor Schneider's lecture, I found Dr. Min, Dr. Schneider worked together for many years, but I found the topic was distinctive and unique. So I think we could understand, the broader context of the interaction between environment and health. So would you please join me welcoming Dr. Min Kyung-duk with a big hand.
Thank you. Can you hear me well?
Yes. Thank you. Please go ahead.
Hello. Good morning and good evening. I appreciate Dr. Chay's kind introduction, and also I appreciate Dr. Kim and Dean Han for inviting me as speakers in this wonderful One Health colloquium. I'm the last presenter, and I know you must be tired, and the lunchtime is coming up, so I would like to cut to the chase. Today, I would like to talk about one of my favorite One Health topic, which is epidemiological studies regarding health benefit from environmental conservation. So current environmental issues like deforestation, air pollution, water pollution, climate crisis, and biodiversity loss are now burning sustainability issue as well. And we need to action immediately for this emergency situation. And I think, I believe epidemiological studies can drive and facilitate the actions we need. For example, in terms of air pollution, there are tons of epidemiological studies which shows the association between the air pollution level and population level health, like total mortality or specific type of disease like cardiovascular diseases. Or there is also epidemiological studies for climate crisis. For example, there are many studies showing the associations between the climate crisis phenomenon like temperature variations or heat waves on health, on population health, like mortality or other diseases. Likewise, I think health studies for forest and biodiversity, especially wildlife diversity, could advocate the importance of forestation and wildlife. So in this regard, I would like to introduce several study examples, which topic is one of One Health topic, and which is also my recent publication. In addition, I would like to suggest several limitations on methodology and in implications, and also I suggest several recommendations for further studies. Here are some of my contents today, and the first topic is about epidemiological study examples regarding health benefits from forests. The first example study is about association between greenness exposure at residential location and their paper's cortical thickness. This research work has been published recently with my team member with various disciplines. The study hypothesis was that people who has higher level of residential greenness exposure, I mean, people living in the greenness area tend to have thicker brain cortex. I thought of this hypothesis based on the previous epidemiological studies showing higher level of residential greenness was associated with higher level of physical activities and higher level of social engagement, etc. And those are related to the higher cortical thickness as well. But there were no relevant study examining the association between the residential greenness and cortical thickness. To test that hypothesis, I collect the data from hospitals. The name is Samsung Hospital, one of the biggest medical institutions in South Korea. And I used the MRI image to extract the cortical thickness of the participant. The medical checkup data also includes the participants' residential location, their home locations, and I use the NASA satellite image data. They shows how green is the specific locations are. So what I did is simply join the two maps and extract the greenness level of the each participant home location. Throughout this process, I generated individual level data set, including both greenness exposure level and their cortical thickness. And I examined the association between the two variables using multivariable regression approach. And as you can see here, the higher level of greenness level tend to are associated with the thicker brain cortex. But the positive association was found only in urban population, not in the non-urban population. The second example study is about association between deforestation and scrub typhus. And this research work was also published recently, 2 years ago. The study hypothesis was that the higher level of deforestation can be associated with the higher scrub typhus instance in South Korea. The hypothesis was based on the previous findings from ecology, and they suggested that the secondary growth of scrub vegetation after deforestation tend to increase density of vectors because the mite vectors or tick vectors like the scrub vegetation environment, which is very stable for them, and which in turn increase the risk of vector-borne diseases like scrub typhus in human population by increasing the contact between the mite vector and human population. I also collect the outcome variable from Korean Disease Control Agency, which is equivalent to CDC in the USA. And I collect the data, district level instance data for scrub types, and I used standardized instance rate for an outcome variable for each district and each year. In terms of this deforestation level, I used the data from Global Forest Change, GFC data, which the data generation process were well described in the Science Journal published in 2013. And they provided global level deforestation information with format of raster type GIS data. So what I have done was counting how many events of deforestation have been detected in certain district in each year to generate the variable for deforestation. So throughout this process, I generated the district level data set, including both deforestation and scrub types. And I examined the association between the two variables using spatial regression approach, which counting the spatial autocorrelations as well. And I found the positive, significant positive associations between deforestation and scrub types risk instance, and the relative risk, the estimated relative risk was between 1.15 to 1.2. My next topic is about study examples for health benefit from wildlife biodiversity, which was topic for my PhD thesis as well. The first example is about association between host diversity, especially the rodent species diversity and the Lassa fever emergence risk in human population in West African region. The study hypothesis was that the higher rodent species richness could be associated with lower risk of Lassa fever emergence in human population. And the hypothesis was based on the previous ecological findings, and they suggest that the activity level of rodent species tend to be lower in the environment of high diversity. So I thought that if the high diversity environment could reduce the activity level of rodent species, then it could also decrease the prevalence among the reservoir rodents, which in turn could reduce the spillover risk from rodent reservoir to human population. To examine that hypothesis, I collected Lassa fever case reports between 2008 and 2017, the period of 10 years in West African region, by systematic review for official and scientific literatures, and also unofficial disease report from news articles and or prominent mayor, the web-based surveillance system. And the area with Lassa fever reports between 2008 and 2017 were regarded as case area, and the area without any Lassa fever report were regarded as control area. And I compared the two regions for their host diversity. And in terms of rodent species richness, I used the data from IUCN, the international organizations for wildlife conservation. They provide spatial range for each species. So what I did is just simply stack them up and counted how many number of species have been detected in certain area. So throughout this process, I generate the area level data set, including both rodent, the number of rodent species and whether there are Lassa fever report after 2008. And using the spatial logistic regression model, I examined the association between the two variables, and I found the significant negative associations, which means the area with lower number of rodent species, which could indicate lower biodiversity environment, could have higher risk of Lassa fever emergence in human population. Next example study is about association between predator diversity and hantavirus infection in South Korea. The study hypothesis was that the higher predator species richness could be associated with lower risk of hantavirus infection, because I found an ecological study which suggested that the higher species richness of predator, which means the more number of predator species, could reduce the activity level of prey species like rodent species, and which in turn could decrease the prevalence level among the reservoir rodent population, and which could reduce subsequently reduce the spillover risk from reservoir rodent to human population. For this hypothesis, I used the data from KDCA, again, that is equivalent to the US CDC in South Korea, and they provided a passive surveillance data for hantavirus infection. So I used the standardized incidence rate of hantavirus infection. The disease name is hemorrhagic fever with renal syndrome in South Korea for each district and each year. In terms of predator species richness, I used the data from National Institute of Ecology in South Korea, and they provided district level information like whether what kind of species have been detected in specific district. So I used that data to calculate predator species richness for each district. So using that process, I generated a district level data set, including both predator diversity and hantavirus infection in human population. And I examined the association between the two variables using various statistical models, including Poisson negative binomials, zero integrated Poisson, etc. And using the Bayesian regression approach, I found the distinguishable reverse U-shaped association between the predator species richness and the risk of hantavirus infection in human population. And this reverse U-shape means there are very low risk of hantavirus infection in urban area, but among the environmentally intact area, if there could be the protective effect of predator diversity on the hantavirus infection. The next topic is about limitations of this kind of study in terms of methodology and implications. The first limitation I'd like to introduce is about diversity of measurement. So for example of deforestation, there are many indicators for measuring deforestation, Actually, what I have done so far is simply using the percentage of wood rent, which is the simplest one. But there are other various ways to measure the deforestation, like edge. The edge means the length of borderline between the forest area and deforested area. Or we can also use the patch density, which or the number of patches, or the interconnectedness between the patches for estimating and for the assessment of deforestation. The diversity in the measurement itself could be problematic in this kind of research, because the research outcome, the result could be different by what kind of measurement they used. So it could be confusing. And more importantly, there are limitations in implications. Although my research work can be categorized as one health topic, because I used the data from various disciplines, and I used various methodologies from their disciplines. But the outcome, study result, implies that we need to protect forest and wildlife because they are beneficial for us humans, not because of their intrinsic value, their own intrinsic value. So this is more like anthropocentric, and which are quite far from genuine One Health concept. The next limitations in implication is about victim blaming, because my research work could be used for victim blaming, because the study result could pinpoint the forest workers who cut trees a lot as culprit or bad guys for the environmental destruction or subsequent public health impact. But there could be real, genuine meaning of bad guys or culprit for the environmental destructions, like big manufacturing companies or capitals behind them. My last topic is about recommendations for further studies. My first recommendation is using the animal health indicators as health study outcome. For example, we can investigate protective effect of predator presence on highly pathogenic avian influenza spillover from wild bird to poultry farms. Actually, these are one of my ongoing projects with researchers in animal health field. And in the preliminary result, I found that the presence of yellow-throated martin in Korean Tambi, they look very cute, but they are very strong predators located in the highest hierarchy in the food web system in South Korea. And what I found it was the presence of yellow-throated martin could reduce the risk of higher pathogenic avian influenza spillover risk in poultry farms, even after adjusting the density of poultry farms or their distance to wild bird habitat. My second recommendation is conducting cost effectiveness studies. And recently published paper from Science Journal suggested that the economic damage from COVID-19 is much expensive than the annual cost for protecting and conservation on forest and wildlife. And they estimated that the cost of COVID-19 is about 400 times higher than the annual cost of conservation. So I think we can apply this approach to local problems. Like we can estimate the cost of conservation in this country, South Korea, and also we can compare it to the economic damages from preventable zoonosis case, and to make to advocate utilities of conservation efforts. Thank you very much. This is my end of presentation, and thank you for your attention. And I'm available for answering questions now and later.
Okay, so thank you very much for your very interesting lecture, which is more, I mean, very relevant to Korean situations. So I believe that many of this audience share the same feeling of mine, that I mean, actually there are many things we can work in the area of One Health, interaction between wildlife, greenness, and human health through his lecture. So, I mean, we have to finish the second session, and I'm very glad for 3 lecturers who finished up this session on time. And especially I would like to thank Drs. Schneider and Wu. They stay up late until like 11 p.m. In your time zone. But thanks to your contribution, I think we could understand that how those ecosystem, wildlife, environment link together with our health, not only humans, but animal health. So I'm afraid because of the time, as I mentioned several times, for those people who have questions, comments, we have a liberty to use the chatting board. And with that, I would like to hand over my microphone to the closing ceremony, Professor Kim Yong-bae. Thank you very much for everybody.
Thanks, Dr. Choi, for moderating session 2. It is really nice to have all the presentation on time. So my bladder was filling up, but I cannot leave the seat because of the exciting presentation. And I think it is almost the time to empty my bladder. As you see, today we had a great and memorable colloquium on One Health in One Medicine, and also One Environment. Personally, it was a great learning opportunity to deliberate the 3 axes of One Health, the human, animal, and environment that we need to consider for our sustainability. Also, I reaffirmed that we should put more coordinated, interprofessional, and transdisciplinary, as well as global effort to address the issues challenging our futures. I want to share some of the slides. As Dr. Han mentioned, in the opening address, this colloquium was the outcome of collaborative effort for the education and research between 3 colleges in Seoul National University: College of Medicine, Graduate School of Public Health, and College of Veterinary Medicine. And I do believe that more valuable outcomes like multidisciplinary course or degree program are being produced. And this colloquium may not be possible without the contribution from many people. I just want to recognize them, including organizing committee from the 3 colleges, chaired by Dr. Ho Jae-han, co-chair Kyung-ho Choi, and committee member Dr. Kang-seok Choi, Dr. Sung-kyun Kim, Dr. Yong-baek Kim, Dr. Jungpyo Lee, and Dr. Hansang Yu. Also, 6 speakers who shared their insightful view and data was indispensable for the colloquium. Especially, I want to recognize that they made colloquium possible by sacrificing their after hour. One last but not the least, I want to give special thanks to Ms. Hewan Seo, who prepared brochure and booklets and processing all other administrative work. She was the key personnel to make this colloquium possible. So we want to give all the big hands to the organizers, speakers, and staff members. And on top of this, I want to express my sincere gratitude to all the audience who attend this event during the work hour. Among the audience, I want to recognize a special attendee. I don't know, I cannot find right now, but he was in the beginning. Dr. Holly Ockwell, who is a DBM and research scientist of One Health lead in the International Vaccine Institute, which is right next to our Ghana campus. I hope this is an informative and exciting session for you all to think about the current status and future perspective on One Health approach for global sustainability and serve as a foundation to stimulate the interprofessional effort. Now I want to wrap up the colloquium. Thank you very much, and I wish you all have safe and healthy days and wonderful night for the speakers. Thank you very much.
수고 많으셨습니다.
네, 고생하셨습니다. 감사합니다.
네, 수고하셨습니다.
네.
네,
수고하셨습니다.
수고하셨습니다.
제가 연락을 못 드렸는데 뭐 감사합니다. 너무 좋은 발표해 주시고
감사합니다.
정확하게 시간 지켜주셔서 특별히 감사드립니다.
우리 민 박사님은 보건대학원 졸업생이십니다.
네.
네, 좋은 기회 주셔서 대단히 감사드립니다.
민 박사님 지금 지난번에 우리 PK 통계학도 강의해 주셨어요. 특별히 감사해요.
네, 감사합니다.
예, 저기 좋은 자리 마련해 주셔서 감사합니다. 저희 보건대학원 BK에서도 저기 이렇게 잘하진 못하겠지만 의미 있는 행사 만들어서 저기 모시겠습니다. 우리 원장님도 이제 그렇게 하기를 바라고 계시고요. 한번 저 모범을 따라서 열심히 해보겠습니다.
감사합니다.
저 학장님 또 교수님도 감사합니다.
Thank you, Dr. Wang, for your contribution. I think it is time to go to bed. 참석하신 모든 학생분들에게도 감사드립니다.
네, 고맙습니다. 또 다음에 더 멋진 프로그램으로 다시 한번 모시도록 하겠습니다. 감사합니다.
네, 콜로퀴움 종료하도록 하겠습니다.